So what I'd like to do this morning will be perhaps a little different from the way we did the panel discussions yesterday. Our first panel is focused on the practitioner's view of microgravity research. We've pulled together a set of panel members who have expertise with respect to ISS National Laboratory research from quite a few different perspectives. Our first speaker will focus on NASA and its use of the ISS and biological experimentation, primarily focused on the benefits that can be recognized for further exploration with the human presence, but also some research that's been done that has focused on ground and terrestrial application. We also have a speaker with us today who will talk more about the rooted ground-based application, if you will, more specifically in clinical care. And we also have two speakers who have, quite frankly, not necessarily considered microgravity as a part of their research endeavors until just recently and are now participating with cases in some experimentation. So pretty diverse panel. I think it will give us quite a bit of insight with respect to the clinical practitioner perspective of how microgravity can be of benefit. What I'd like to do is have each one of them present a brief presentation related to their topic of expertise, and then we'll assemble a panel for some Q&A. So our first speaker this morning is Dr. Julie Robinson. She is the Chief Scientist for the International Space Station. In this role, she represents all of the ISS research disciplines at the highest level in the program and provides information and recommendation about the ISS as a scientific research platform to not only senior leaders but decision makers who are critical in establishing research programs. I want to divert from her bio just for a second to say that we at CASIS, and we'll explain this a little bit more, are focused on how we can take the benefits of microgravity research and apply that towards terrestrial application. Julie brings to this a valuable set of expertise where she can actually identify the work that's been done in the past where the intended outcome was not always for terrestrial application, but more for space exploration. But she does a fantastic job of helping us translate those benefits to where we can recognize those on the ground. Julie has overseen the transition of the ISS from the assembly period when, if you can imagine, the laboratory was being built and research was trying to be conducted, to just recently in the last number of years where it's gone to full utilization and over hundreds of currently active investigations are happening at any point in time. She has an interdisciplinary background in the physical and biological sciences, and her professional experience has included research activities in a variety of fields, including virology, analytical chemistry, genetics, and field biology, along with remote sensing. She has a Bachelor of Science degree in chemistry and a Bachelor of Science in biology, both from Utah State University, and she earned her Doctor of Philosophy in Ecology, Evolution, and Conservation Biology from the University of Nevada, Reno in 1996. She joined NASA as a civil servant in the Office of the ISS Program Scientist in 2004 and was named Deputy Program Scientist in 2006. And we now have the great opportunity to have her serving as the Chief Scientist since 2007. So, Julie?
Well, thanks so much, Twain. My remarks are going to be very brief, but what I wanted to do was really try to take the deeper information that Joel just provided you and pull it back to what the space platform means as a research opportunity for biomedicine. And so, let's see if I hit the right button here. Nope, let me try that one. Okay, now it's working. So I want to start with droplets, which was in Joel's presentation, but I want to remind you of the way that we talk about water to our freshman biology students, right? Freshman biology students, we tell them water is the most important thing, and its properties define the properties of life. Life is all about surface tension forces, controlling water, having water move in the places it needs to be. And so when you think about it, a droplet like this, which has surface tension forces deciding how it's going to relate to the leaf, and then gravity pulling it down, mashing it flat, and so you get a combination of those things in a photo like this one. Of course, when you go into space, surface tension forces become the dominant force, and you don't have the gravity squishing effect. I think you'll like gravity squishing effect better than the differential equations that Joel showed you, maybe? I was giving him a bad time about that last night. And so here's a picture of Karen Nyberg playing with her drink bottle. It's almost the obligatory photo if an astronaut's on the ISS and doesn't get a photo like this for their memoirs, they've done something wrong. But what it really shows for you is the difference for fluids. Now take any eukaryotic cell, anything with a soft cell membrane, and what's going to happen in space is that cell in culture by itself is going to also do the same thing that droplet just did. And so in fact, if you take a cell that's squished flat, to some extent by gravity, its shape is regulated by the interactions of its membrane proteins, its membrane charges with the surfaces, with other cells and so forth, and you get a structure to that. Put that into space, and if it's in free culture, it will become spherical if it can in any way. And you see accompanying with that all these changes in the way that the cell works. So this is an experimental system to push a cell in a way that you can't do in any other place. And maybe that's all well and good. It sounds like a gee whiz thing to do, but it really has fundamental opportunities for understanding things like cell division. So here on the left, you see one of the simplest eukaryotic cells we can study, Saccharomyces cerevisiae. And on the left is a normal budding process going on in those yeast cells on the ground. Normally, those yeast cells are going to bud at the same point every time. So you see the little small one in the upper left, where the bud's coming off. And you never see on those cells a bunch of bud scars because they always do it in the same direction. You can find one if you look at it at the right point in time. On the right, you see yeast cells that have divided in space, and you can see that cell has divided roughly 7 times, and every time it divided in a different place. So what this tells us is there is something about cell division that is different if you take gravity out of the equation. And in fact, when we look at a variety of model systems, we see that is translating into the things that we care about very much from a biomedical perspective, like cell-cell interactions and like cell division and differentiation. And here's an example, if I can get this to, let's see. There we go. So this is an older experiment, but it's a rotating wall bioreactor in space and on Earth. And what you're seeing here, if you look at the top, those are the ground controls for the experiment, and you see those cells stayed in suspension pretty much. But on the space controls at the bottom, you see those cells adhering. Well, these are prostate tumor cells. And when you do the histology after you bring the samples home, and unfortunately these samples never came home. This particular experiment was on the Columbia flight and was destroyed in the reentry, but we have the video that came down. So we never got to do histology on these. But when you do histology on those tumor growths, they look more like they do in vivo and less like they do normally in vitro. And so that's a really compelling, but it's almost anecdotal. This is new work, and that's one of the reasons that CASIS has had a stem cell opportunity out there, because how you build on this result and how you learn from this to really understand what cells are doing when they divide and what tumors are doing when they form and spread is a completely open book to the scientific community. Now, as Joel mentioned, you can take that, jump up from the cellular level, so don't get whiplash. We'll go straight to a human body here, and you see that lack of a gravity vector affecting everything. Neurovestibular, cardiovascular, because they're not working very hard, floating around. Bone, because those muscles that aren't working very hard aren't providing the right feedback to the osteoclasts and osteoblasts. Muscle, because they're not working well. The immune system, probably because of structural and motility, other issues like that. Nutrition, because the food doesn't taste good, because radiation is knocking things out. Behavior, because these guys are locked in a can for a long period of time together. And of course, the radiation environment. So you have all of those things together added on top when you look at the systems level, look at a whole organism. And so we have 2 major systems that we're using on the space station to really work at this higher level. One is a mouse system, and that's getting ready to fly for its first time later this year. And we'll have pretty much every SpaceX launch is going to have rodents on it that are being used for a variety of studies to bring this up to the systems level. This is one of the studies we squeezed in during assembly that was done by Amgen, looking at the an osteoprotein inhibitor and helping to understand the mechanism of the way that osteoprotein inhibition works ahead of their development of prolia. And other kinds of discoveries, things like the optic disc edema, which we still don't have a complete understanding of why that's caused, and so those kinds of systematic issues we can look at. And some things that we're doing as we shift to the crew member as a subject rather than mice as subjects are we will have one-year missions starting. The first one will be in 2015, and there'll be periodic one-year missions after that time. If you look at the years of the past one-year mission and the nationalities of them, there have basically been four guys who have flown more than 12 months in space continuously, and they all did it prior to 1999. And if you can think of what you could study and what you could measure in patients in 1999 compared to what we can study and measure today to understand the effects of all of those processes I talked about, you can see why doing one-year missions today and using both the Russian and the US crew members becomes so important. On this first one-year mission, we also have the unique situation in that Scott Kelly, who will be the first US one-year crew member, also has a twin brother, Mark Kelly, who will stay here on Earth. And so that offers some unique opportunities to do genetically matched work, really look at genetic differences, and look at what happens following the one-year spaceflight. And then the final real advance or change I want to talk about is the transition from the NASA-supported research from a single PI model to an open source science model. And so if you see here, if you start kind of on the lower left blue box, in the past we started with one investigator who competed in a research announcement. And then once they were selected, NASA figured out how to do the hardware and how to get it launched for them and so forth. And then they went to the space station, that's number 3 in the top. And then their samples come home. And then number 5, the PI publishes it. And if the journal requires him to put it in an archive, he does, or she does. And so the new model is one aimed at really trying to get those discoveries out to a broader community, not just the club of people who happen to get selected as PIs, but really let the comparisons of what kinds of gene expression, protein expression, metabolomics, all of those things to compare what space looks like to what we're learning not space looks like, really to integrate it into the broader sphere of understanding of genomics in the world. And so here you have a science advisory team that sets some standard conditions for a lot of experiments. NASA figures out how to fly it and puts it in the right hardware. And when the samples come down, it goes into an open bioinformatics database that's linked to the standards of practice for other Earth databases. And then there's the opportunity to get NASA funding for data analysis. There are also independent outside data users. For example, pharma can just go browse it and see if they can find something useful, see if that means they should do an experiment in space, perhaps. And cases can select users. And so this is going to be a real strength in the way that the next 10 years of life sciences research on the space station is functioning. And I put this chart in primarily for those who might be interested in tracking some of this omics type work. We are at the stage of having the science definition team get together and start setting some of those parameters, setting the plan for the plan. And over the next 2 years, this will be finalized and shaped so that we're sure that we have a system in place that really serves the needs of the broader biomedical research community. And so with that, let me move along, let you keep it rolling. Okay, you want me to stay here? Yeah. Okay.
Thank you, Julie. Our next speaker is Dr. Robert Hamilton. He is the co-founder and chief scientific officer of Neural Analytics Incorporated. Dr. Hamilton's research focuses on cerebral dynamics in several neurological conditions, including TBI, subarachnoid hemorrhage, and normal pressure hydrocephalus. Dr. Hamilton focuses on advanced morphological features of intracranial pressure and cerebral blood flow velocity to predict future pathological events, including reduced cerebral blood flow and intracranial hypertension. Dr. Hamilton earned a BS in biomedical engineering from Washington University in St. Louis, and he completed both his Ph.D. and M.S. degree in biomedical engineering at UCLA. Dr. Hamilton.
Great, thank you. Thank you very much, and thank you for everyone coming out today. I think it's amazing to me to be here talking about space research, because a year ago I was mainly focused on, I was in an intensive care unit at UCLA looking at traumatic brain injury and not really understanding how this research would apply to space-based conditions and the advantages of that. So again, I guess I have a disclosure. I'm a co-founder of Neural Analytics. It's a company I founded following my Ph.D. So a lot of my Ph.D. work was related to, again, traumatic brain injury, subarachnoid hemorrhage, and looking at intracranial pressure. So for those of you who don't know, intracranial pressure is a really important metric as a prognostic factor for outcome. So as the pressure increases, whether that's from a bleed or diffuse swelling, if you don't take care of that, I think I'm getting some feedback, then it can be deadly. So we looked at that a lot during my PhD and in my lab. Unfortunately, one of the only ways to measure ICP reliably is to drill a hole into the patient's head and measure it directly. Now, in the US even, 40% of those patients that need ICP monitoring don't get it because of a lack of expertise. I do a lot of work in Africa where there's just no opportunity to measure intracranial pressure. So once I met the Cases team and Dwayne and Cindy, I started thinking about the opportunities of performing some of this research on the ISS and looking at the changes in physiology in 1G and then in microgravity. So this has been a well-known problem for decades, that this microgravity environment can lead to papilledema that Julie mentioned, and some of the visual changes in the facial edema that has been seen over the last few decades. So this kind of fit right into my wheelhouse of changes in cerebral blood flow, changes in vascular resistance, venous pressure, and so on. So what has been done is transcranial Doppler is a useful technology that was developed in the 80s that has been used and is currently being used on the ISS to gauge the physiological or the physiology of these astronauts. The nice thing about it is non-invasive, but unfortunately a lot of the results so far have been inconclusive for estimating intracranial pressure. So where my expertise comes in is signal processing and machine learning type applications. So what we did was we said there's a lot more information in that signal, and so we extracted many more features out of that signal and then start to look at how to classify different physiological conditions. Now in this case we'll be looking at intracranial pressure. So these are some of the initial results from our UCLA study that were done against the invasive monitor. So you can see there in the green line, this is traditional transcranial Doppler measurements, somewhere around 59% for predicting intracranial hypertension, to where some of our advanced features were able to get an accuracy of 92%. So we can go into some of the physiology of actually extracting physiological information out of this waveform signal, because at the end of the day, we want clinicians on Earth to be able to say, this subtle change in this waveform means something. And that's why we're so excited about working with the ISS team and trying to extract some of that information. So again, I've spent a lot of time looking at some of the Earth-based conditions, pseudotumor and cerebral malaria in particular, because they are related to some of the venous pressure changes that do occur in microgravity. So keeping this relatively short, again, I'll be on the panel to ask questions, but we're looking at more advanced features, and we're really benefiting from this idea of open collaboration, because we actually get to utilize some of that information that is being collected right now on the ISS. So we are directly benefiting from this new collaborative model that NASA and CASES are putting together. So thank you.
Thanks, Robert. Stay up here. We'll just have the panel kind of listening. Thank you. Our next presenter is Ms. Gwen Owens. She is currently a dual M.D. Ph.D. student in the UCLA-Caltech Medical Scientist Training Program. She's currently pursuing her Ph.D. in biochemistry and molecular biophysics at Caltech in collaboration with Dr. Pamela Borkman. Ms. Owens graduated summa cum laude from Cornell University with a BS in biological and environmental engineering and in biology. She was instrumental in the design, development, and current experiment, which is currently on orbit on ISS, a study of Huntington crystallization and microgravity. This experiment, the first Caltech experiment to take place aboard the ISS, was just recently launched on the last SpaceX mission to the ISS. So Gwen, thank you for joining us.
Thank you very much, Dwayne. I wanted to start the presentation with a video that CASES made that describes our experiment.
Oh, gee, that's great. Is the lighting halfway decent? Yes, indeed. They've got the flag up now, and you can see the stars and stripes.
When I watched Neil Armstrong step on the moon, I was amazed that I could sit and watch TV and then walk outside and see the moon.
0 and liftoff of the Gemini.
I always dreamed of being an astronaut, but I never thought I'd be an astronaut actually. The mission for both of my shuttle flights was to contribute to the assembly of the space station. Now that the space shuttle's retired, our job at CASIS is to find innovative ways, creative ways to maximize the utilization of the space station, which will ultimately benefit mankind. ARC 1, Advancing Research Knowledge 1, is our first mission going aboard the space station. The ARC 1 suite includes physical sciences, biological sciences, and education payloads. One of the experiments that we're really excited about is an investigation into Huntington's disease, which could lead to new treatments for that disease.
My lab studies the three-dimensional structure of proteins, and I'm studying a particular protein that's called Huntington. And this protein, when it's mutated, causes Huntington's disease. When a patient has Huntington's disease, it's essentially a death sentence. And often their children will also have Huntington's disease because it's a genetic disorder. Our group and other groups have been trying to crystallize this protein for almost 20 years. We've done maybe 100,000 different conditions of this protein, and nothing has really worked up to this point. There are trillions of these protein molecules that are floating around in solution, and slowly over time, these protein molecules start to come together and start forming what's called a crystal. For our technique to work, every protein needs to look the same in every part of the crystal. There's been some really good evidence that crystals grow bigger and more perfect in microgravity. On the ISS, we really hope to be able to finally get these crystals that are big enough and perfect enough that we can solve the structure of this Huntington protein. Not only is the ISS such an international effort, but this research experiment is as well. It's this consortium of universities, private companies, and the government all coming together to try to make this experiment happen.
The International Space Station is complete. 6 astronauts and cosmonauts are doing research, are advancing knowledge, and benefiting both human exploration and mankind here on Earth. Join CASIS as we embark on this very important mission to advance research knowledge aboard the International Space Station.
So it's been known for many years that microgravity can help crystals grow larger, grow more perfectly. This is an example from a paper from the early 2000s where a protein grown on Earth and then grown on microgravity, you can see in some instances is 10 to 20 times larger. In addition, the quality of the crystal is much better as well based on some of these other metrics. And you can see that crystals grown on Earth versus microgravity actually have different properties as well. And so my lab is a crystallography lab, and we've been working on this Huntington protein and trying to crystallize it for many, many years at this point. And so we were very excited when we heard the CASIS had a request for proposals to try to crystallize proteins that are very challenging to crystallize on Earth on the International Space Station. So we chose to use a device that's called a handheld high-density protein crystal growth assembly, which is a crystal growth chamber that had previously been used on space shuttle experiments where crystals were grown. In this apparatus, we put a little bit of this Huntington protein that we're trying to crystallize in one chamber, and we put a solution that helps it crystallize in a separate chamber. And they're kept separate until they reach the International Space Station, at which point the experiment is activated by astronauts, and crystals can start to form. So this is an image of the lockers that they go onto on the International Space Station. And we finally launched these experiments on April 18th, so right about a month ago. And they were activated on the International Space Station 3 days later. So these experiments are currently ongoing. This is an image of our research team and 2 other research teams that were setting up protein crystal growth experiments at Kennedy Space Center prior to the launch. I'll take any questions on the panel. I wanted to keep this brief, but thank you very much.
So our final panel member this morning for this first panel is Dr. Cliff Dacso. Dr. Dacso joined the faculty at the Baylor College of Medicine in Houston in Texas in 1978 as an instructor of internal medicine. He received dual appointments there 2 years later as a clinical assistant professor in the Department of Medicine and as an assistant professor in the Department of Microbiology and Immunology. Dr. Dacso later moved to the University of South Alabama and became there the chief of both the General Internal Medicine Division as well as the Infectious Disease Division, and then later vice chairman of the Department of Medicine. In '88, Dr. Dacso joined the University of California at San Diego, where he served as the chief of general medicine and geriatrics, as well as chief medical officer of their outpatient VA clinic and the director of the internal medicine group. Dr. Dacso rejoined Baylor in 1992 as the vice chair for clinical affairs in the Department of Medicine and the chief of both general internal medicine and international medicine. He became the vice chair for strategic development there in 2000 and assumed his current position at Methodist and the University of Houston in 2005. Dr. Dacso is a colleague working with CASIS. We have recently created a collaborative where we are going to endeavor in omics-based research, and we're very fortunate to have you speaking here today. Thank you.
Thank you. Thank you very much, Dwayne. That's a very generous introduction. I'm sorry my mother wasn't here to hear that. My mother would have been so proud, my father wouldn't have believed it. So I'm here to talk about molecular biology on the ISS. I was asked to tell you everything I know about molecular biology in 5 minutes, and it was a stretch because I probably only know about 4 minutes worth. But and then the other stretch, of course, is being the last speaker to try to say something that's even slightly original. We have begun our collaboration with CASIS about 9 months ago, initially working in the area of intracranial pressure, which Dr. Hamilton just described, and then subsequently in the area of molecular biology. What's really fascinating is that space, as Dr. Hamilton has described, and how you've also heard in other talks this morning, is richly endowed with data. And the big challenge is to mine those data in such a way as to make meaningful terrestrial applications. So as I reviewed the literature, I was amazingly surprised to find the amount of information that was available having to do with spaceflight. Here, for example, is a set of curves describing worldwide non-commercial space launches, and you'll see that there's a very tight correlation with sociology doctorates awarded in the United States. A very meaningful association, who would have suspected? And further, there are even further curves that are extremely well correlated, showing that the U.S. spending on space science and technology correlates very strongly with suicides by hanging, strangulation, and suffocation. Who knew that there was such a rich amount of data to be generated from spaceflight? What I want to talk about today in about 3 minutes is what we're looking at now in terms of the molecular biology of circadian rhythm. As you know, circadian rhythm is a strong determinant of many biological functions in virtually every organism from yeast on up. You heard a little bit about C. elegans worms. C. elegans worms have genes that control circadian rhythm. And when you get into eukaryote populations, the genes that control circadian rhythm are almost perfectly conserved in evolution. So they've got to be pretty important, and they really are. And what we found on terrestrial applications is that the circadian rhythm genes indeed control not only the sleep-wake cycle, but also have dramatic effects in terms of immune function, and even some of the proteins have a dramatic function in terms of the survival of cancer cells as they seek to metastasize. A specific one that we've looked at in our laboratory has been SRC2, steroid receptor coactivator 2. SRC2, as some of you in the cancer world know, has a central protein in the effect of both estrogen and progesterone on cell function. What our laboratory has been able to do is create a knockout model of mouse with knockout of SRC2. And in this slide, what you see, and I'll walk you through a relatively complex slide. This is from a recent publication in Cell Letters. LD is light dark, DD is dark dark. This is wild type, and this is SRC2 knockouts. And what you see here is when you take a mouse and entrain them to light dark cycles, because they are nocturnal, they are active almost exclusively at night. They are active exclusively at night. You turn the lights off and they start running on their little wheels. You put them in a full day, night-night environment, 24 hours of night, and they still do sleep because obviously the proteins have been coded. But the circadian rhythm, even though the sleep duration is exactly the same, the circadian rhythm does tend to shift. If you take the SRC2 gene, the steroid receptor coactivator gene, and knock it out in this mouse, what you find is they become free running. They lose their ability to control day-night cycle, and they are exercising randomly. This is an extremely interesting observation, and so we've decided to take this into space for you know, you can put a man on the moon, but you can't control the slides. Let's see. Sorry, yeah. I'm sorry, this is another slide from that same publication showing the functions, the biological functions that are contained with, that didn't go, did it? I see it, but you don't. All right. I can't put it, oh, I can't, and I can't change a slide. Even though I was the AV guy in high school. Well, it was, no, let's go back to the, well, it was the guy who couldn't even make trainer on the football team, got to be the AV guy. But we all went to medical school. There we go. Okay, thank you. So this is a heat map from the same experiment looking at knockout mice at, and this ZT10 is just a particular time in the circadian cycle. And again, wild type on the left, SRC2 knockouts on the right. And what you see in this heat map, those of you who are not familiar with looking at heat maps, red is enriched and green is downregulated. And what you see is that there's an almost exact inverse in some of these very critical biological functions and processes. And these are the processes that we've identified that our SRC2 knockout circadian rhythm determined. So if you look at this, the biochemistry here, you'll see that there's just an extraordinary range of chemical functions anyway. So there's just to wrap up, there's a lot of other things you can do in space. I didn't know much about tardigrades before. Tardigrade is this really interesting little macroscopic critter that lives on algae and plankton. And what has been found, just to know the remarkable things that you can do in orbit, is that, and this was published just recently, is that these little critters can survive for 10 days outside of the space station. You put them out there, they dry up, and then you bring them back in and just water them a little bit, and back they come, as if just as elegant as they were before. And so, of course, NASA is now taking that type of information and is going to make them into astronauts, although, again, with this picture, it's unclear why the tardigrade actually needs a spacesuit. So they are now doing even further experiments, and this is from the most recent ISS mission, where you see the little tardigrade floating outside with his little sunglasses on and his little glass of umbrella drinks. And so when you ask, what are the types of experiments that you can do in the ISS, they range from doing knockout mice experiments with circadian rhythm all the way to umbrella drinks outside the space station. Thank you very much.